people think that it's on our street that what's coming. Well, I'm looking at that. But it was interesting.
All right, so we are down to our last lecture. And so what we're going to do today is we're going to finish up the eye. We've got half the class hasn't done the lab exam, so I'm going to wrap up a couple of minutes early, let them go, kind of regroup for the lab exam. We have a review scheduled for next Tuesday. Let me know, I wasn't quite sure if you had a preference of start time. I have it currently listed at 10. I have to be gone by 11:30, so I can move it up if you think you're going to need a little bit extra time. We can start it at 9:30. I wasn't quite sure if you guys had preference or you guys are so done you're not going to tell me.
I think it'll be enough time.
Okay, 10 to about 11:30.
Yeah, I think it'll be enough time.
Okay, so 9:30. Say again? 9:30. You want 9:30? I think. Okay. What I will do is I will get here at 9:30, right? I will be here at 9:30, and then I can just keep going. So as people are coming in, we can start with their questions. You know, we may double up a couple of times on different questions, but we'll do that. So we'll start at 9:30, and just keep going until whatever. Is that okay with everybody? And so you don't have to show up at 9, you can show up at 10, you can show up at 10:30, whatever. I have to leave by 11. That's the only thing that's going on. Okay, so did you guys have any questions over ears and eyes so far, the things that we've covered so far? Okay, so, all right, so we talked about how sound travels, right? And we talked about properties of sound. What are the two properties of sound that the nervous system codes for? Pitch. Pitch, right? Is it a high-pitched noise or is it a low-pitched sound? And we said this dealt with frequency of sound waves, right? The frequency of sound waves. So we had a diagram that looks like this. Okay, which is going to be the high-pitched sound, blue or purple? Purple, right? Because it has the higher number of waves going past a particular point, cycles over time. Okay? What was the other property? Intensity, right? How loud? How loud is the sound? Okay? And that was reflected in the pressure difference, right? Between the peak of the wave and the trough of the wave. Okay? So, how does the nervous system code for intensity? It's code, yeah, it's decibels or units, that's great. And how does that affect the transmission of electrical signals to the brain? The hairs, right? The hairs are going to get hit, right? So, tell me about action potentials. Number of action potentials over time will increase for loud sounds. Okay? Now, tell me about pitch. Right? We have a picture of how pitch was coded. Action potential sent to the temporal lobe. And what's key about where would you send a low-pitched sound? Lateral side, the low frequency area, high pitch, medial side. All right? And that's because when we talked about the basilar membrane, remember the membrane on the bottom being displaced? We said for high pitched sound, that occurs in the cochlea closer to the end. right? And low frequency sound waves will cause this tipped end of the center of the snail, right, to be displaced. And then you send those action potentials to a particular region of the temporal lobe, right? Low pitch is going to be lateral, high pitch, medial side of the temporal lobe. All right? You guys okay with that? All right? Why do we send action potentials to the inferior colliculus? Auditory stimulus, reflex response, right? And then it travels up to the thalamus to our temporal lobe. Okay. Next, we switched over and talked about utricle saccule. What is detected in that region of the inner ear? Stopping and going, right? Acceleration, deceleration, linear. Where do we detect rotational motion? Semicircular canals. Okay. And what are otoliths? Yeah, little calcium rocks, right? And they move at a different speed, right? It's just like driving with rocks in a car. They will move at a different speed than you do and bend hair cells. Right? And we said that was important because we have mechanical receptors that are activated that way. All right, we talked about Meniere's disease, right? Why does Meniere's disease have signs and symptoms of tinnitus? What's tinnitus? You're hearing things, right? And why is that an effect of this disease? It affects the inner ear. Yeah, it's going to affect the inner ear, the hair cells. We're going to create action potentials that are inappropriate, right? Why do you get vertigo? Semicircular canals, we're going to trigger inappropriate electrical activity coming from the semicircular canals. So you will perceive that your head is spinning in space. Okay. Another thing that can cause vertigo is where otoliths can become dislodged. Right? If you have otoliths that are floating around in your inner ear, what's going to happen? It's going to feel like you're moving. Right? And that's another thing that can cause vertigo. Okay. You guys good on ears? Pretty straightforward? All right. So then we started talking about vision. And we talked about light coming through the eye, crossing the cornea and the lens. What do the cornea and the lens have in common? They bend light, right? They bend light. They're transparent. Light will pass through, but the light will get bent. And you remember we talked about what's the amount that the light is bending? What is that called? How do we reflect that? Refraction, right? And we talked about the refractive index, right? And we said, does the number get bigger or smaller when you bend more? Larger the number, the larger the bending, right? Your shorter your focal length, okay? We talked about two different conditions. We talked about myopia and hyperopia. Myopia and hyperopia. With myopia, right, is the image focused in front of or behind the retina? In front. It's focused in front of the retina. So hyperopia will be behind, and then that tells you how you're going to have to correct, what type of lenses you would use. So what type of lens corrects for myopia? dead center in the retina. Light comes straight in your eye. Blind spot's a little far down. Fovea centralis. What's important about the fovea centralis? Most accurate vision. Visual acuity is the greatest. So, these sit around the periphery of the retina. Are these going to be used for accurate vision? No, they're the sides, right? They're the sides. These cannot see in color. They are colorblind. So these give you black, white, gray vision. Okay? Gray vision. They function best in dim light. Okay? So now that we actually can be outside some, when it's not snowing, right? What I want you to do is some evening go sit outside on your deck or outside on the sidewalk somewhere, get in a nice lawn chair and a book, whatever, drink. Sit there and watch the sunset. Don't look at the sun directly, that's bad for your eyes. After it sets, can you see color?
Yeah.
Not usually, right? Once that sun goes down and it becomes dusk, right? You're familiar with dusk, right? Do the leaves look green? No, they look gray. Does your grass look green? No, it looks gray, right? Trees, gray. Flowers, gray. Everything's gray. You've lost the colored vision. What cells are you using now? Rods, right? Because they function best in dim light, right? And so once that light goes down to a particular point, you lose color vision. You need bright light to see colors, right? If you ever want to know what the color of a shirt is, right? Navy, black, really hard to see. You have to go put it in bright light to tell the difference, right? Colored vision, bright light, okay? Dim vision, or dim light, it's all gray, right? You lose the ability to distinguish colors, okay? Cones, right? C, what's C going to stand for? Color, right? Cones, color vision. These are centrally located. So what are we going to have at the fovea? Cones, right? Function best in really bright light. All foveal cells are cones, all of them, okay? So cones, you've got a couple of different kinds because you've got multiple colors, right? So red, green, and blue cone cells, right? Red, green, and blue cone cells. There is a sex-linked disease, red-green color blindness, right? Where it's usually in men, where men do not have one of these receptors, and so red looks green. You cannot distinguish between the two. Have you noticed what they do to stoplights? Do you ever notice? You guys need to go look at stoplights. All right? So after you've set out with your drink, looking at the sunset, right? You're going to go drive down your street until you come to a stoplight, right? I want you to look at the green light. They have colored them blue now. So they are blue-green. Why is that important? So you can't see green and you can't see red. What do you know if you're supposed to do with the light? If the red light is green and the green light is red.
All the way home.
Yeah. Yeah, the order's always the same.
Yeah.
The order is always the same, but the green is now green, blue. Because you can tell, if you're colorblind, you can tell blue from red. You can't tell green from red. Okay? So we've got multiple types of cone cells to determine the different colors.
I have a first question. If you can't tell like green and blue, it's really hard for them to differentiate from them. So if you hand them something that's green or blue, you have to like tell them. So what would that be?
Right. You can lose these different. It's not always. The green-red color blindness is the most predominant. Right. So that's the most common in men. But there are other cones that you can lose that will blur the wavelength. All right. So this is kind of a big picture. The names of the cells are actually based on their shape. This one looks like a cone rod. Okay? And inside these cells you have these discs which contain visual pigment. Right? Visual pigment. And these are the pigments called opsin, opsin molecules. And this is what's going to bind the light. It binds to photons. So we've got all this pigment stacked in these cells. These cells are then connected to our bipolar cells. Right? When light comes in, it's got to travel through, and it will actually bind to the pigment in these cells and activate them. Right? Now, when this binds light, it actually becomes chemically converted. Remember how we chemically convert things? So we talked about how you can take like acetylcholine and break it in half and make it 2 new chemicals. Here we're going to chemically convert our pigment when it binds a photon. Okay? Now that's kind of important because you have to chemically convert it back before it can bind another photon. Are you okay with that? So you've got to change it back before it can bind another photon. Alright, so you have now sat outside in the dusk, watched the sunset, gone down, driven down your street with the stoplights. Right? Now you're going to go to the movies. Right? You're going to watch a movie. We're going to do this at noon. Right? So when you walk out of the theater, you're blind. Right? Right? So what's happening at that point is that your pupils are now dilated, so you have tons of light coming in, right? What type of receptors have you been using? Rods, right? But now we're in bright light, and we have just completely saturated all of our receptors with light. They have to reset before they can bind again, and that takes time, right? So that's kind of where you get this whole, you have to reset those before they will bind to light again. All right. What vitamin is supposed to be really good for eyes? Okay. K and A. Vitamin A, rabbit, carrots. Okay. That's because that's used to make this molecule. Okay, so now what we need to do is talk about the fovea, right? Because we said that's really important. It's where you have your most accurate vision, right? The fovea centralis. It is smaller than the head of a straight pin. It's very, very tiny, right? And for most of the retina, there is much convergence of areas of information. And we've talked about convergence of areas of information before when we talked about the nervous system. And the example that I gave you was when someone has a heart attack. So when someone has a heart attack, typically you feel pain. Where do you feel pain when you have a heart attack? Arm, right? Is your arm actually in pain? No. We said that's caused because the arm and the heart actually lead into the same pathway, So they converge, right? Convergence of areas of information, right? And so that when that actually travels down the pain pathway from the heart to that tract, you think it's from your arm because normally it's your arm that you're hurting, not your heart, right? Now that convergence also occurs in the retina, okay? So you are going to have lots and lots of cells feeding into one pathway, right? I wrote the ratio down. So there are 130 million receptors for 1 million ganglion cells. So we've got 130 million receptors. Right? So we're going to have 130 receptors feeding into one ganglion cell. Right? How do you know which cell receives the light? You know? Right? It could be any one of those 130. right? Is that going to give you accurate vision? Yeah, you're gonna have no idea, right? And so someone said, one of those kids is your child. One of the 130. Go find them, right? You can't do that, right? That's not a very accurate way, okay? That's what we have. We have 130 different cells feeding into one ganglion cell. That is an inaccurate way to see, okay? Now, the fovea, most accurate vision, greatest visual acuity, has a one-to-one relationship. One cone receptor cell, one ganglion cell. Okay, so periphery out on the sides, you don't have that. 130 feeding into one. Fovea, one receptor cell, one ganglion cell. Incredibly accurate, right? You know, a photon hits that cell because it travels down one ganglion cell that is responsible for monitoring that one receptor. Okay? So that is why you have that greatest visual acuity. The other reason is that, I don't know, I don't have a picture of it. Nope. All right, so the other reason is, you remember when we were talking about, first of all, it looks like a little dimple, like it was an indentation, right? The reason it is an indentation is all the ganglion cells are pushed to the side, right? So that the light can actually travel directly to the rods without having to cross through all those layers. So it's actually, it's kind of like where you part your hair, right? That fovea is a part in the retina where you're going to separate those ganglion cells and pull them to the side so the light has more direct access to the cones of the rod. The cones of the light. Okay? All right. So you guys good with why the fovea has the greatest visual acuity when light hits that region? One-to-one relationship between the receptors and the ganglion, and the other cells are separate, pushed to the side so light has better access. Okay? Okay. Retinitis pigmentosa. Okay, retinitis pigmentosa. This is a disease that affects mainly rod cells. Okay? So what I want you to do is I want you to ponder rod cells, what they do, where you find them, and come up with some symptoms that you could predict based on knowing that this disease affects rods. All right, what do you guys got? Night blindness. Okay, that's one, right, and that's absolutely correct. You cannot see at night if you have damaged rod cells. What else? Where are rods located? Outer. Right? Right? And what's that called? Peripheral vision. So you have tunnel vision. You can see things that are directly in front of you, so you cannot see to the side. Right? And so that's absolutely correct. So retinitis pigmentosa affects rods. Two things that you would expect with this: tunnel vision and night blindness. Right? Will be affected by, will be caused by that disease. Alright, you guys, you want me to write that down for you? You guys got that? Got it. Alright. Visual pathways, alright? So now what we're going to do is talk about what happens when a photon binds to a receptor. Okay? This is going to begin at the photoreceptors, right? We have a photon being bound by pigment either in a cone cell or a rod cell. It's going to end in the visual cortex of the cerebral hemisphere. Do you remember where the visual cortex is located? Back. What's that lobe called? Occipital. So it's going to end in the occipital lobe. Right? The message crosses two synapses before it heads towards the brain. So we're going to talk about a little bit about those. And we actually did mention it, right? Bipolar cells, so photoreceptor to bipolar, bipolar to ganglion. Right? So those are our two synapses we're going to cross. Our ganglion cells have axons that are going to come and become bundled together and converge on the optic disc, right? You remember the optic disc? We said that's where you have no photoreceptors. You have no photoreceptors there because you have, right, all those axons coming out. And then you have blood vessels as well, right? The optic disc where these axons will penetrate the wall of the eye and proceed as the optic nerve, right? 2 optic nerves, 1 for each eye, will reach the optic chiasm. You remember what the optic chiasm was? Crosses, okay? Right? Now, what we're going to do is we're going to look at how those axons are bundled. And what it really is you know, are we looking at things on the sides, right? If we're looking at in our left visual field, how is that information coming into the eye's process? Okay? And you have to remember that light travels in a straight line, right? And it'll make a little bit more sense. I'm actually going to show you the picture of this because I think it will be better when you look at a picture about how we're going to bundle axons when we look at things that are in our side vision. Okay, so here's our picture, right? What I'm going to do is get you oriented. Here are the 2 eyes. Your nose would be here. Okay, everyone okay with this? 2 eyes, nose here. We're looking up, right? Because like you're lying flat on your back looking at the ceiling. Okay? Now, what I want you to do is I want you to focus on looking at something that is in your left visual field. So that's going to be this light blue over here. Everyone see that? Okay. Now, can you see the dotted lines? Right? So I'm looking to my left. Okay. I've got light rays coming in straight, right? And some are going to enter my left eye, and some are going to enter my right eye. You guys okay so far? Okay. What I want you to do is I want you to take and trace that line. Right? So it's got light coming in. Is it hitting the lateral side or the nasal side of the eye? Nasal side. So we're going to travel down. See where it's crossing sides? Right? What's that called? Optic chiasm. Okay. Now let's do the other eye. Got light coming in from our left side. Where does it hit? Nasal side? Lateral. Right? So let's take a look at that axon bundle. Did it cross sides? No. Stayed on the right side. So things from your left visual field end up on the right side, right? Even though you got 2 eyes, 1 on each side, right? The left information, the nasal side, crosses sides. Okay? That bundles together, goes to our superior colliculus. Why are we sending visual information to the superior colliculus? Reflex response to sight, right? So it's got to travel there. We've got projection fibers. Projection fibers were the ones that travel to different areas of the brain, right? Going to come into the right hemisphere, okay? If you want to do the right field, right? We hit the nasal side on the right eye. We hit the lateral side, temporal, right? That side doesn't cross. This side crosses, ends up on the left side, goes to the occipital lobe, the visual cortex. Okay? Things that you see in the center, right, coming straight in, they will end up on both sides. You guys okay? Okay. Now, your visual cortex, right? Visual cortex, occipital lobe. Okay. We've done the motor homunculus and the sensory homunculus. You remember those? Right? So, you remember things about kind of the face and the hands. So, tell me what you know about the eye. Where would you want most of your visual cortex dedicated to as far as the retina goes? Where do you collect most of your information from? Most accurate information? Foveus centralis. So what that visual cortex is it's going to be a map of the retina, but you're going to have more of it dedicated to the fovea, where you have the most accurate vision. Does that make sense? Yep. Okay, so someone has a pituitary tumor. It's producing visual effects. Okay? what do you know about the anatomy of the optic chiasm that might explain that? You have a pituitary tumor. It's gonna grow, enlarge in size, right, and push on the optic chiasm, right? And once you start crushing that cross, right, you're gonna start producing visual effects. It's gonna muck up the electrical signals traveling through there. Okay, that is it. All right. All right. Do you guys have any questions over this information? Pretty straightforward, I think.